In this video, we will be finding a protein of interest, manually mutating its amino acid sequence, and comparing that sequence against a bioinformatic database to find other proteins of high sequence similarity via the BLAST program. We will also be looking at any possible protein-protein interactions using the string database and viewing the function and structure of our protein. First, we need to choose a protein of interest. Go to uniprot.org. The protein should be involved in a pathogenic mechanism. For demonstration purposes, I will be using the urease subunit alpha protein found in Helicobacter pylori. You can search your protein in the search bar located here. Hit enter. On the UniProt entry for your protein, you will find a table of contents to the left. You can click on these links to bring you to the respective section. Here, you can find the function of the protein. You can find information such as interactions, and you can find other information such as structure and sequence of your protein. Here, we want the sequence of our protein of interest that we will be manually mutating. Click on the sequence section. Copy the first 50 to 200 amino acids of the protein sequence. Paste the sequence somewhere that can be saved, such as a Word document or a text file. It might be helpful to download the FASTA file for your protein sequence, like so. This will make it easier to copy your protein sequence. Once you have done that, replace all the alanine amino acids with glycine. That's all the A's with G's. Using our new mutated sequence, go to web.expasy.org/blast/. Here we will be comparing our sequence against a database of known proteins to see which proteins our sequence is most similar to. Enter your sequence in this box. Ensure that uniprot KB/swissprot only is checked off. We want to search against this database only. Then, once you've done so, hit Run BLAST. This may take a few seconds to a few minutes, depending on your sequence. Once your page has finished loading, you'll see on the left side a quick access table. Find list of the matches and click it. This is a list of proteins that are similar to your sequence, with the most similar sequences situated at the top. Now, via the quick access table, find alignments and click it. Here, you will find detailed information about how your protein sequence was compared to the hits under List of Proteins. You will find the E value, bit score, and percentage identity here as well. The E value is a statistical indicator of how similar your sequence is to the sequence listed. The lower the E value, the more likely it is that the sequence is similar to your protein sequence. Bit score is similar. The higher the bit score, the more similar the sequences are, and percentage identity is a percentage of the sequence that is similar to your sequence. In this case, for demonstration purposes, I use the exact amino acid sequence from my protein without manually mutating the alanines to glycines. Go back to the list of matches. Click on the accession number here of the most similar hit. In other words, the topmost hit. This link will bring you to the UniProt entry for your protein. UniProt is a database consisting of curated protein descriptions. This page for your protein will contain information such as function, structure, and protein-protein interactions, as I mentioned earlier. Clicking on the interaction link will bring us to the interaction section of the protein, detailing known protein-protein interactions. Similarly, clicking on structures will show any known or predicted structures for the protein. Do note, it is possible that your protein you have chosen does not have any predicted structures or known protein-protein interactions. These entries are curated, and if there is nothing under these sections, it is possible it has not yet been discovered or analyzed or entered. If your protein does have a structure section, you will be able to click on one of the PDB hyperlinks, which will bring you to the PDB entry for your protein structure. PDB is a database for protein structures. You will notice that there are multiple hyperlinks which bring you to different PDB websites. These PDB websites share their data. For example, PDBJ is the Japanese PDB website, and PDB is the European one. We will use RCSPDB. Here, you can download PDB files. PDB files are text files that describe the 3D structure of the molecule or protein and are generally used in programs. You can also find the 3D View tab on this page which will bring you to an interactive representation of your structure. You can turn off components and even find structure motifs using this tool. Going back to the UniProt page, go to Structure and scroll down. Here you have hyperlinks to other 3D structure databases, the first of which, SMR, stands for Swiss Model Repository. Clicking on the hyperlink will bring you to the SMR page for your protein. Similar to PDB, you will find information on the protein on the left and an interactive protein structure viewer on the right. Next, go to string-db.org, also known as the String Database website. Hit Search. Here, you can input your protein name, which you can get from the UniProt page, and the organism name, which can also be found on the UniProt page. Then hit Search. Ensure that the correct organism and protein are selected, and continue. This website shows the interaction network between your protein of interest and other proteins in that organism. You can click on the lines to bring up the interaction information, or the proteins themselves for information on the protein. Scrolling down, you'll find the predicted functional partners. And, to the right of it, an associated score. Generally, a higher score means a closer interaction network between your protein and the predicted functional partner. In my case, URI H, F, and B work closely with URI A. That concludes this video. You should now be able to blast an unknown protein sequence against a protein database to find similar proteins. Then, by accessing the UniProt page of those proteins, find more information such as structure, protein-protein interactions, function, and more. Finally, you should now also be able to use string to observe for your protein's interaction network.